Let us now enter into our discussion of proteins. The material in the next couple of lectures will underpin all of our discussions of protein function as well as enzyme mechanisms. So this is perhaps some of the most important background content we're going to cover. The next two lectures will cover amino acid side chain structure and chemistry, and so you should know that you're going to be required to know all of the amino acid side chain structures, and you're going to need to be able to interpret them in terms of their chemical properties, most notably hydrophobicity and ionization state or polarity. We're also going to talk about proteins as polymers, and that's probably their most important role is as large biopolymers. And this is going to lead into a discussion of protein secondary and tertiary structures. So let's begin. Amino acids, free amino acids, are essentially polyprotic acids or bases. They have multiple ionizable groups. They all have at least two ionizable groups. That is the alpha amino group and the alpha carboxylic acid group. This is sort of a complicated diagram. This is just a titration curve. As we've seen previously, we see that there are two pKa's. The pKa to the lower left is our acidic pKa, obviously. pKa of around between 2 and 4. That's that carboxylic acid group. And then we have a higher pKa that undergoes an ionization or protonation, deprotonation rather, around pH 10, and that's our alpha amino group. This titration curve just kind of underlines one of the most important features of amino acids, and that is how their ionization changes as we change the pH. Now, let me say that for the most part, we're not going to be talking about amino acids as free single amino acids, but this is an illustrative case. So this happens to be the amino acid alanine. If we look at the upper right structure there, we see that there's a CH group, the central CH group. That's our alpha carbon. And adjacent to the alpha carbon, there's a methyl group, right? That's our R group. We're going to see that R group can be one of 20 different possibilities. Also attached to the alpha carbon to the right are the alpha carboxylic acid and the alpha amino group. And if we look there at that upper rightmost corner of the titration curve, we see that the amino group is shown as deprotonated, therefore uncharged. Remember, it's a Bronsted-Lowry base, while the carboxylic acid group is negatively charged, deprotonated. Remember what this means. Remember how we interpret this, right? We are way above the pKa of the carboxylic acid group, right? Something like 8 orders of magnitude above the pKa in terms of hydronium ion concentration, and therefore low proton concentration, mostly deprotonated and charged, obviously. Whereas we are above the pKa of the amino group as well, way out there at pH 11 or 12. That means, again, it too is deprotonated, but in the case of the amino group, the base, deprotonated means that it is uncharged. Now, as the pH drops, or you could think of it differently as we change the equivalence of OH in our solution, of hydroxyl in our solution, then we see that at the other extreme, down at pH 2, now we're below the pKa of both of those functional groups, meaning what? Well, the concentration of hydronium ion is high. Both groups are protonated. In the case of the Bronsted-Lowry base, the amino group is protonated and charged. The carboxylic acid is protonated and uncharged. Interestingly, for the amino acids, these multiple ionization or ionizable groups, we also can think of a pi, an intermediate state in which both functional groups exist as ionic in their charged state. They will refer to that as a Zwitter ion or Zwitter ion, right? Both the amino group and the carboxylic acid group are charged. Now, this is worth pointing out. Again, we're not going to talk about free amino acids ever again after today, but it's worth pointing out that in general, these amino groups and these carboxylic acid groups do pop up in amino acid side chains, and it's important to remember that at physiological pH, they're both charged. In the case of the base, protonated, charged. In case of the acid, deprotonated, charged. Okay, that is something that we need to just be familiar with and able to bandy about off the top of our heads. So think about it. What else to say about amino acids? They're chiral molecules. So that alpha carbon is a chiral center, and you remember chirality. The rules that I'm giving you here are straight out of your organic chemistry textbook. I encourage you to go back and familiarize yourself if you need to. Reminding you here too that your text, Leninger, as well as an organic text and a molecular biology text, are on reserve in the library for you. So we rank the substituents of that alpha carbon, that chiral carbon, according to their atomic number. The lowest atomic number points away from us, so that proton, that alpha proton attached to the alpha carbon, is facing away. And we see that they go counterclockwise in this way. So there's the amino group, that's our highest order, the carboxylic acid group next, and then the R group, in this case, again, the methyl group at the top. So there we have a counterclockwise rotation, that is to say left, or sinister, if you will. If we were using the RS nomenclature, you'd say rectus for right-handed, sinister for left-handed. In nature, the L form predominates. We'll hear about violations of this as the semester goes on, and you'll hear more about it next semester to a certain extent, but by and large, we're talking about L configuration amino acids in biology. So the next several slides are just a sort of a who's who of the amino acids. Let's start with the simplest amino acid, glycine. Now I just told you that amino acids are chiral. That's not true of glycine, and you can immediately see why. There's not one alpha proton, but two attached to the alpha carbon. So because we have equivalent functional groups there, we don't have a chiral center. It's worth remembering. It's also worth noting that R group for glycine being a proton is going to mean it has some special properties in terms of configurations that it can adopt, right? And we'll see that steric hindrance by that R group plays a big role in structure formation. Something else to remember here is that there are three things in terms of naming nomenclature to remember. There's the common name glycine. There's the three-letter version of that name, glycine, GLY, and then there's the one-letter code, G. You'll have to learn these as well. And if we look at alanine on the right side here, we see that methyl group is our side chain, as we've seen previously. This, of course, is a chiral molecule, and ALA is a three-letter code, and A for the one-letter code. Some of them make perfect sense. Others require a little bit of thought to understand why. We might refer to them by a particular letter. Now, polar amino acid side chains. Let me back up and say that alanine with the methyl group as its R group is kind of a nonpolar side chain. It's a small nonpolar functional group, but it is nonpolar. Glycine, we might say, is neutral in terms of hydrophobicity of the side chain with a single proton. But let's think about the polar amino acid side chains for a few minutes. These are side chains that we don't normally think of as being charged, but we've also already spoken about how situations in which they might become charged. So let's just walk through these. On the left, cysteine, we see that now instead of a methyl group, we have a methylene attached to a sulfhydryl or thiol. This has a pKa of about 8. That sulfhydryl deprotonates around pH 8 or 8.2. So again, if the ionization state at physiological pH 7, we're below the pKa, therefore protonated, therefore uncharged in this case. Serine hydroxyl, we've talked already about the pKa of serine. That hydroxyl deprotonates almost never under normal circumstances, so we'll say it has a pKa of greater than or equal to 13. Similarly, for threonine, we have a very high pKa. Threonine, very similar to serine, as you can see, just an extra methyl group coming off of that beta carbon. So again, there's some nomenclature for you. The alpha carbon is that central carbon, beta, gamma, as we move out. Another interesting detail about cysteine is its ability to form a disulfide bond. That is to say, it can dimerize with another sulfhydryl, so both of them will lose a proton, and instead they'll share a bond between the two sulfur atoms. So we'll see this as a common structural motif in some proteins, and I'll point it out when we see it. I guess I should also say that these proteins are more common in the oxidizing environment exterior to the cell, and maybe in some organelles. But by and large, we'll think of the cytoplasm as being a reducing environment, and therefore this sulfur will be considered to be reduced and protonated. So frequently we don't see heavily disulfide bonded proteins in the interior of the cell, although there are plenty of exceptions. Now, another set of polar amino acids would be the charged amino acids, and of those, let's first consider the acidic side chains. Aspartate, also known as aspartic acid, I'm not a stickler about this, but I guess formally aspartic acid would be the state in which that carboxylic acid on the R group is protonated. Aspartate would be the conjugate base, which I've drawn here. Note that D is the one-letter code. Glutamate, or glue, has an extra methylene. That should be CH2 at the alpha, beta, gamma carbon, and then again at acidic side chain. I'm not a stickler about these pKa's. Let's just remember that there are about 3 or 4 for those side chains. So immediately, let me remind you that although we're not going to be seeing a lot of charged alpha amino and alpha carboxylic acid groups in protein polymers, those side chains now, their protonation state becomes what we care about. So D and E are acidic side chains, and we can amidate those side chains, and they're therefore no longer acidic, but they have an amino group there. And there are 2 versions of them as well, and you can sort of see where those names come from. Asparagine from aspartic acid, acin or in for its one-letter code. Again, add an extra methylene at the gamma position, and we'll see that becomes glutamine, glen, or Q. So those are 2 that are a little bit difficult to keep your brain around, but you have to memorize them. Also, polar side chains. We're going to see these side chains playing an important role in protein. One moment while I plug my computer in. We're going to see these side chains playing important roles as both hydrogen bond donors and acceptors in various kinds of chemical reaction mechanisms. So that's an interesting point to remember. Charged amino acids, the basic subset. So lysine, arginine, and histidine. Consider first lysine and arginine, or lys and arg, or K and R. Lysine's pretty easy, just 4 methylenes and a primary amine out at the end. And I've shown this in its protonated state, charged state, of course. Right? I'm again reminding you physiological pH. We're well below its pKa, which for lysine happens to be around 10. For arginine, somewhat higher, maybe 12. Again, I'm not being a stickler about the exact numbers, but 10 for lysine, 12 for arginine, close enough. For arginine, we see that what kind of functional group do we have here? A guanidino group. Histidine, far right. We've already mentioned the fact that this molecule can be protonated with a pKa of somewhere around 7. So again, protonating that second nitrogen and becoming positively charged. I've mentioned before, and I'll mention it again now, histidine is an important side chain because of the position of its pKa, which is fairly malleable, so we can imagine pushing that protonation state around quite a bit around physiological pH just by changing the nature of the chemical environment in which it resides. And that'll be important because histidine is going to play a role both as a proton donor and a proton acceptor in a single chemical reaction, enzyme reaction mechanism. So those are our basic side chains. We got 3. Remember our acidic side chains, we got 2. No point in belaboring this, but here again is a similar slide for glycine showing you how we go through these protonation states. I'd like you to, on your notes now, why don't you practice sketching a protonation or a titration curve for arginine and for aspartic acid. So remember, in those cases, now you'll have 3 ionizable groups, and you should be able to draw a similar curve to the one on the left here. So we'll go over that in class as well, but it's worth working through those just as a way of practicing thinking about protonation of side chains. Let's see. Interesting chemistry about the polar and charged amino acids is the side chain reactivity. So primary amines, carboxylic acids, and sulfhydryls are all going to play important roles in reaction mechanisms because of their reactivity, and I'll leave it at that for now. But let's move on to nonpolar amino acids, our other class of amino acid side chains. These are a little bit harder to recall, but you can just at a glance see why we call them nonpolar. You see lots and lots of carbons, methyls and methylenes. Some are straight chained, some are branched. There's a sulfur atom in methionine, but it's not considered a terribly polar sulfur atom. So valine, Val, or V, right? There's a branched side chain for you, branched at the beta carbon. Leucine, also branched, but branched now at the gamma carbon. Likewise, isoleucine branched at the gamma. An important thing to remember, not confusing leucine and isoleucine is very easy to do. Just remember that they're both branched, but leucine is branched at gamma, isoleucine branched at beta. And methionine, straight chain with the sulfur at the alpha, beta, gamma, delta position. Okay, interesting cyclic amino acid, that's proline. So you can see here now that the alpha carbon the side chain emerging from the alpha carbon actually loops back around, forming a covalent bond with the alpha amino group. This is referred to as an imine, perhaps inappropriately. I'll let you go look at your organic chemistry text and tell me whether you think that's an appropriate usage of that term. Nevertheless, it's a cyclic side chain, and what I've drawn for you here is the population of the peptide bond in all the other amino acids and with proline. So this is leaping ahead a little bit, but I'm asking you to remember that amino acids are linked together via a peptide bond, an amide linkage that links the alpha amino group of one side chain with the alpha carboxylic acid group of another. So you can see there, two alpha carbons linked together through this peptide bond. And the trans position is favored in 99.9% of the time for all the other 19 amino acids, but favored in the trans position 95% for proline. Now, I wanted to insert this here, and hopefully not too confusing at this point. We're going to come back to cis-trans isomerization, or the structure of the peptide bond, but it's worth pointing out here what the relevance of proline in protein structure. It partly evolves or devolves from this issue of cis-trans isomerization. We don't get free rotation around that bond. That isomerization is a difficult reorientation, and so if we find ourselves locked in one configuration, then we might have to do some significant rearrangement that might come at a higher energetic cost. So let me give an example, which would be protein folding. Normally, protein folding, as we'll see in subsequent lectures, can happen very, very quickly, but can be significantly retarded by the adoption of perhaps a wrong configuration of proline at one amino acid. So a protein that would otherwise fold quickly might get trapped in a trans when it should be in a cis configuration, and the whole molecule then has to reorganize itself. So can dramatically slow down things like protein folding. But there are other biological significances to the cis-trans isomerization in proline. And let me introduce now a little bit of relatively recent scientific literature. So why do we care about cis-trans isomerization? Why do we care about structural change in this cyclic amino acid side chain? Well, this is a model of the serotonin receptor. Let me back up and say, well, why do we care? Well, the biology of a system can be reduced to chemical change. So what are we looking at here? We're looking at the serotonin receptor, one of the serotonin receptors. We're actually looking at a monomer of the serotonin receptor. So if you can imagine, you probably all know what serotonin is. It's a neurotransmitter. I've drawn it for you up in the upper left, also known as 5-HT, 5-hydroxytryptamine. I think you can all maybe see why it would be called 5-hydroxytryptamine. If you can't, stare at that structure for a minute and figure it out. Okay, this is a complicated slide, and it's demanding that you leap ahead in your understanding quite a bit, and I hope that you'll bear with me. The serotonin receptor sits on the surface of a cell membrane, perhaps a synapse, and it sits there as the 5 members in the organization. So it would be a pentamer. So for each one that we're looking at here, say there's one monomer of the pentamer, and so you'd have to use your imagination to think about what the full receptor might look like. So 5 serotonin molecules binding at 5 different sites, and sort of what the cartoon on the left shows you is in red are some of the amino acid side chains that interact directly with the serotonin. The thing that we want to point out, though, is that little blue blob sort of in the middle. That's a proline that bridges 2 helices. As we'll see, prolines do not form normal secondary structures in proteins very easily, but that's not the story here. The story here is that binding of serotonin to the receptor results in a conformational change, so there's a big idea and term for you to think about, results in a change in the shape of the protein that ultimately causes a change in the cis-trans ratio of this particular proline. So in the unbound position without serotonin, the receptor sits in the trans conformation. The proline sits in the trans conformation. When we bind serotonin, we switch from the trans to the cis conformation. That change precipitates a very large reorientation of that little green loopy structure, right? I'll call it a loopy structure for now because we don't know what protein secondary structures is, but some of you are probably aware that that's an alpha helix. And without regards to what a helix is, you can see that the green ribbon on the left reorients dramatically on the right with respect to that other, the gray helix. So the point here is just that ligand binding, serotonin being the ligand of the serotonin receptor, ligand binding reorients that helix, called the helix M2. That reorientation does what? It opens the channel, right? So this is actually, the serotonin receptor turns out to be an ion channel. So when you bind the neurotransmitter at the receptor, the ion channel opens up, ions flow across the membrane, depolarizing it or polarizing it, I can't remember which right now, but in any case, there's a signal that's transmitted that changes the concentration of ions across the membrane. If this is a neural membrane, then we're talking about the transmission of a neural signal, right? All boils down to the cis-trans isomerization of one proline. This cartoon is looking down that channel now. So now let's imagine that we're serotonin, we're floating outside the membrane. We're looking down at our receptor on the left. We're waiting to bind at the receptor. And then when we do bind our receptor on the right, the proline isomerizes from the trans configuration to the cis, and pop, you can see the pore between all of those ribbons opens up, and now ions can begin to flow down that channel. So this is a model, I should say, and I don't want to go too much more deeply into this. Again, I'm sort of belaboring this example of the importance of proline, published not too long ago in Nature, where they were able to actually observe how the binding of serotonin affects the cis-trans isomerization properties of this amino acid. And if you're interested in the paper, you're welcome to read about it, and you can present your